Hello friends, welcome you in Research Units. In this video we are going to see folic acid-induced acute renal failure model. So for that we will go ahead. In this slide we will see allocation of the animals, and these are the groups. Around 6 groups we have separated. In that all the animals are divided in normal control, disease control, reference control, and treatment, higher, medium and lower doses. And in this day we will see how to induce using folic acid. For induction we will prepare 150 millimolar sodium bicarbonate solution. In that we have to add 250 mg as per the dose preparation, 250 mg folic acid, and in single dose IP we will give the all animals G2 to G6. All groups we have to give the doses, and these are the reference control. We will take ibudilast 10 mg per kg, and we have to dissolve it in 35% polyethylene glycol 400 per oral. And our drug dose may be, it's depend on you how what is your drug and what is its per kg human dose, and as per your dose you can take your drug concentration. We will go next slide. In this we will see the study design here. From first day of animal procurement, we have to keep the animals for 7 days in quarantine, and then we have to acclimatize the animals for 5 days in laboratory condition, where we are going to conduct the experiment. And meanwhile we have to conduct randomization of all animals, and then we will allocate in 6 different groups that we have seen in previous slide. And day 1 we will start dosing with our drug. It's up to you. You start before 7 days or 1 day, or after the drug induction. It's depend on your study model, because it's a acute model, so we will conduct this experiment for 7 days only, or it's depend on the drug induction, how the folic acid while inducing the animals, and how what is the disease level it occurs at that time. It's depend on that. We have to standardize the model first, then we will go further. So we have to, even we can induce it day first also, and first we have to induce and then we will start treatment. That type of experiment we can also do. So here on day 2 we will induce the folic acid, and we have to prepare the folic acid 150 millimolar sodium bicarbonate solution. In that we have to add the 250 mg per kg folic acid. While we are preparing the solution, sodium bicarbonate solution, and we are adding folic acid, it's showing milk like color, milky orange like color it's showing. So we have to dissolve it completely for 10 to 15 minutes up to it cure transparent water like color, orange water like color, while preparing this folic acid, because sometimes it is not dissolved properly, so induction of the disease will not occur. So this is how we have to start induction. We have to do the induction, and second, meanwhile we can start our dosing too. Same day we can give dosing after 1 hour or before 1 hour we can do the dosing or treatment. We will continue that time, and it's after 2 days. While we are doing this type of experiment, we have to standardize the model first, and in standardization of model we have to see that sometimes what happened, the disease will recover after 7 days. So in that condition we have to terminate this experiment in 2 days. So while we are inducing, and we have to check that what is the parameters of their blood parameters after 24 hours or after 2 days. So after 2 days we will check serum creatinine and serum urea. On that we can conclude that what is the disease means how much it is induced, how much it is induced, what is the disease induction level. So by that we can confirm that, means it will work or not, it is recover or not. After 7 days we have to again, during standardization process, we have to check it is showing induction or not. If it is showing induction, then we can continue this model for 7 days. Otherwise we have to do this experiment for 2 to 4 days. That is more than enough. After induction, now after, if we are doing the same model here for 2 days or 4 days, then after induction, one day after 24 hours we will collect the urine before termination. If you are going to terminate it 7 days, so on day 6 you have to add all the animals in metabolic cage. Before adding the all animals, we have to check the, we have to give the doses of drug treatment, and we have to 2 times we have to give water, double from their dose. 2 times water we have to add, so that it can produce required volume of urine. And before this necropsy, we have to collect their blood, and in that we will check the burn and serum creatinine level, serum urea, and sodium potassium. And the body weight we have to do twice a week. And after necropsy we will take wet organ weight to calculate relative organ weight. And finally we will collect after 1 days, after 24 hours completion in metabolic cage, we will remove the animals and collect their uh, volume. Actually we have to record the urine volume, and it will be useful to calculate in the burn clearance and creatinine clearance and glomerular filtration rate. So for that we have to collect blood urine. And finally, in parameters, in blood urine we can do kidney marker test KIM1 and NAG model, and also we can perform gene expression RT-PCR, NFR2, and QO1, HO1, ILF1 beta, TNF alpha, NF, NF kappa B, and inos, nitric oxide synthase. And finally we will do kidney histopathology, and in that one kidney we will add in the complete in histopathology. By, we have to cut this kidney in the transverse section, full kidney, one kidney, and one kidney we have to divide in 3 pieces. One piece we will use for gene expression, and one piece again we will add in histopathology, and one we will keep for another parameters if anything needed in future. Now finally we will see how to calculate creatinine clearance, burn clearance, and glomerular filtration rate. To calculate, we will use this formula for creatinine clearance. Here we have added, means 1000. Why we have added that we will see in later. 1000 into urine volume into concentration of creatinine in urine and concentration of creatinine in blood, means serum. And second, burn clearance, urine volume into concentration of urea nitrogen in urine, and concentration of burn, means serum. So creatinine clearance and burn clearance were further calculated per hour by dividing in creatinine and burn clearance by 22. Means we will collect and drug for 24 hours, means what urine volume for 24 hours, and for per hour we have to, now we have to calculate the burn clearance and creatinine clearance and glomerular filtration rate for per hours. So we have to divide these whole volume by 24. So lastly we will calculate GFR, means creatinine clearance, and burn clearance mean we will collect, and we will get the estimated GFR. When urine volume in ml, urine concentration of creatinine UN are in millimole, means urine urea and creatinine concentration in millimole, whereas in blood concentration of creatinine is micromole and blood concentration of PUN is millimole. So here it is micromole, that is why we are adding here 1000. So this is how we can calculate and conduct whole folic acid induced acute renal failure experiment. How was the experiment explanation? Please give your comment, and if you have any doubts, we will clear it by the comment. Thank you for watching Research News. Thank you very much.